So you sit just alone here.
Yeah, biology. Yeah.
Some of you might know Nikolay from the focus period that we had. We bumped into each other and started talking about this ancient DNA, and it sounded so fascinating. So we wanted to hear more about it. So very welcome, Nikolay.
Thank you very much. Thank you very much. It's a pleasure, great pleasure to be here again. So I had the honor and privilege and pleasure to meet some of you in May, a few months ago, during the elite period. And it was very interesting. It was a very interesting experience. I learned a lot, and I realized we have a lot of common interests. Although I'm sitting at the biology building, and you're sitting here, well, it's not far from each other, but it was not obvious to expect that we have so much common interests. Yeah, at that time, we talked a lot about cells. But today I'll talk a little bit about ancient DNA, something else. But I hope you guys can get equally excited about ancient DNA field as I am, and we can have a nice discussion. So please interrupt me and ask questions because it's important to have a common language. Because I know for sure I'm from biology department, but I'm originally from theoretical physics, so I defended my PhD thesis in theoretical physics, and then after a few years working in physics, in 2011 I switched to medical genetics because I met people from this building in Malmö, which is called the Clinical Research Center. So they're clinicians seeing patients, diabetes and cardiovascular diseases, a little bit of cancer. And I started working with them. So it's a medfac, it's a Department of Clinical Sciences. So there are three centers that medfac has in Lund. You probably know there is a BMC, Biomedical Center, there is Medical Village, and the third one is a Clinical Research Center in Malmö. So this is how I felt. First year, so I was lost in translation. I literally didn't understand a single word. So it was like white noise for me. So it was white noise every day, every week, every month, for many, many years. And then you start understand 10% of their talks and 20% of their talks, 30%. And it took me a lot. I still don't understand everything, but I'll try at least. I think I have a feeling of what terminology people use in biology and what terminology people with with mathematical background use. And maybe I can translate some words for you, some terminology. So I'll try to watch my language and try to find understandable terminology so that everyone can be on the same page. And then in 2016, I was recruited by this organization. It's called Science for Life Laboratory. If you're not familiar with this, it's an infrastructural organization. So the Swedish government decided to build this institution not as yet another academic institution in Sweden, but as an infrastructural sequencing and life science facilities institution. So that people working in this SciLife lab, they are assisting and helping and supporting research in other academic institutions in Sweden. And I'm a bioinformatician, so this is what I'm doing. I'm basically looking at the sequences. So this is a DNA sequence. This is how sequencing data looks like. So it's very different from the analysis that people do. Maybe, well, I can say about physics, for example. There people would talk about atoms, about molecules, but not really about sequences. So bioinformaticians are specifically looking at the codes, at biological. So they're looking at biological molecules, but they're not interested in 3D structure, for example. They're not interested in interactions. They're interested in the sequence and how the sequence leads to certain properties of our body, of our cells. Whether we can get diseases, whether we can get some certain talents, for example, So all these so-called phenotypes, they can be linked to our DNA in theory. And this is what I'm doing. This is what bioinformaticians are doing. And then step by step, I started I got interested in evolutionary science first, in pure biology. So to say, less medicine, but more pure biology. And then I got interested in ancient DNA, and it started in 2014 when I met Svante Pääbo. It's because my boss at CRC, Ulla Hansen, from the Department of Clinical Sciences, he was actually a friend with Svante. And so they had personal friendship, and their families, they meet and see each other. So this is how I started collaborating with Svante. And
Svante to the right.
No, that's me. That's me. That's me. So he is more bright.
In the other photo.
In that, oh right. So you meant this? Yes. Got it. Yeah. No, that's a Neanderthal. So I hope everyone knows the Svante is a Neanderthal guy.
Some percent of him.
Right. So and then so we started. So what we collaborated on was sort of evolution of skeletal muscle, gene expression in human skeletal muscle in comparison with great apes. So this kind of research, and also following up this in Neanderthals, Neanderthal DNA. This is the type of research we were doing. And then recently, well, 2019, I sort of started working sort of on a daily basis with these two guys, Louis Dahlen and Anders Gutterström, from Naturhistoriska Riksmuseet, the Swedish Museum of Natural History. And those are great guys. They're doing paleogenetics research. Louis Dahlen is a mammoth person. So he works with mammoths, mammoth genomics, mammoth ancient DNA. And Anders, he's a human. He's no, I mean sorry for my language. He's concentrated. He's concentrated on ancient humans. No, that's a big deal actually. In evolutionary research, it's very important to sort of set the boundaries, because evolutionary people are sensitive. They see a huge difference between working with human DNA and non-human DNA. I think they're exaggerating, but that's why I'm sort of describing it in this way, very carefully emphasizing that this is a mammoth DNA and this is a human DNA, because for them it's very important for some complicated reasons. I can talk, but maybe not now. Yes, so, and also with Anders, we were doing a lot of research on ancient human microbiome. So it turns out that you can actually extract microbial DNA from archaeological samples. And if it's some interesting microbes, for example, pathogenic microbes, then it tells you how these people lived, what kind of diseases they had in the past. That was super cool. And this is this kind of research we were doing. And I would say this ancient microbiome, it's my specialty now. It's in ancient DNA, it's probably the topic that I know the most. But here, I'll give you some sort of brief introduction. And I had a dilemma, how deep should I give some overview, or should I concentrate on a particular topic? So I decided kind of to mix a little bit. But I still hope it will be interesting for you. At least I will try to explain it in an interesting way. So what is ancient DNA? It turns out that you can sort of extract DNA from the nuclei, from dead cells, from ancient tissues. So you have archaeological samples, and then if you look at their cells, they're dead, but there are still nuclei, and you can extract, you can crush this nuclei and extract DNA. DNA is well preserved in contrast to RNA. So RNA is digested very quickly. So there are special enzymes that chew it up very quickly. So there is no concept, or almost no concept, of ancient RNA. But ancient DNA is well preserved, and also ancient proteins. Proteins are also stable, so they survive. They don't degrade with time.
Can I ask you, is this under lucky circumstances that it has been hidden from sun and or is it always like this?
Indeed, a good question. So basically, yes, DNA degradation depends on temperature. It depends on preservation condition. So basically, in Africa, it's very hard to analyze ancient DNA, despite the human being came from Africa. We know very little about the very, very ancient humans. And this is for this reason that DNA is almost always degraded. That's why now paleogenetics community, they're moving to proteins. So they start looking at proteins more and more, because proteins survive better than DNA in Africa. But in permafrost, in Siberia, for example, you can sort of even extract preserved RNA. So not only DNA. DNA is very well preserved in permafrost. And even sometimes RNA. So that's sort of the grade. When you're moving from north to south, you would expect to find less and less, fewer and fewer well-preserved ancient DNA samples. And up north, the preservation conditions are great. The problem is that there were not so many humans living there. Only mammoths, maybe. Always cave lions, maybe bisons, animals, but not so many humans. Yes, so and you sort of read the sequence. So there are special machines that can read this DNA sequence. There are some problems with ancient DNA. It's as I said, very few samples. So there are not so many bones. There are not so many bones with well-preserved DNA. And also, usually it's a tiny amount of DNA. So even if you extract maybe a few sequences, it will not be enough for you to do any meaningful analysis. That's the problem. So in biological sciences and medical sciences, people always, almost always do statistical analysis. So for statistical reasons, you would like to have a large sample size. So because all these assumptions, they are central limit theorem, they are large numbers. You need large numbers in order to satisfy assumptions, and that's why ancient DNA is particularly difficult for statistical analysis for that reason.
So if you find an incomplete molecule, can you figure out where, what part of the sequence that you have been found?
Yes. So so the way we do it, I'll come to this in a few seconds, but just quickly, the way we do it, we read pieces of DNA sequence, and then we have a human genome. So human genome has a reference, so-called reference human genome. It was sequenced in 2006, and now it's even complete. So there are no gaps. So we know exactly the reference human genome. And the reference means it was just a random individual. It's not an average genome of all population, but it was just one individual was taken. The DNA of this individual was sequenced. And now all other samples we find, including these ancient samples, we compare its DNA with the reference. So we need a reference to compare against. And all the mutations, they are sort of detected in terms of comparing it to this particular query samples to the reference. Mm-hmm. So we read short pieces of DNA, and then we align it. So we find the position where exactly this piece came from that gene, or maybe from intergenic region. But the shorter the DNA is, so why is it difficult to work with degraded DNA? Because the shorter it is, the more non-specific you can so the placing of this segment becomes less and less specific. Okay. Because so people know that below 30 nucleotides, so if this sequence is just 30 nucleotides, so it can map basically to mouse genome with 100% affinity. Mm-hmm. So it can map to ape. So it's non-specific. Mm-hmm. You even don't see that it's human. Okay. It can be microbial. But there is no way actually to check whether it came from, if it's short enough, there is no way to check whether it came from a microbe or from a human. Yes. So the sequencing, the way we do sequencing, or not me, but the people in the lab, so they do drilling in order to make sure that no external, no exogenous DNA is dropped in the sample. And then they put this special clothes just to have very clean lab, very clean conditions in order not to contaminate these unique samples. Because the problem is that if you take a DNA from an ancient individual and just place it against your DNA, you will not find a difference. You will not see a difference. It will be identical. So that's the problem. That's why if people here in the lab, they drop their own DNA to the sample, then you will be thinking that you are working with ancient DNA, but it was actually your DNA. And yeah, just a second, if I start talking about this. So the very first paper that Svante Pääbo published, so he's a godfather in this field, because this was absolutely the first paper in 1985. He was the only author in Nature. It was the very first paper that tried to read the sequence of a mummy, Egyptian mummy DNA. So this is how the field started. But then, so this is a sequence that was published. For that time, just to read a few stretches of DNA, what was a breakthrough. But then Svante acknowledged many times that this was whoever's DNA, but not the mummy DNA. It's probably, it could have been Svante's DNA or his collaborators, whoever DNA, but not Egyptian mummy. Because at that time, there was no way, actually. Now we have sort of tools and ways. But at that time, as I said, if you just place this DNA against your own DNA, you will not see a difference. Moreover, if you place this DNA against ape DNA, you will not see a difference. That's similar, yeah. So we actually astonishingly similar.
So the first paper had these big mistakes?
Yes, so unfortunately, but I mean, this is how the field started. That was a proof of concept that you can actually, you can think in this direction. At that time, no one thought in this direction, that you can actually say something about the human past by looking at their DNA. Because we can understand now a lot about how they lived, what they ate, what kind of style of life they had from the DNA. So it was like a paradigm shift. People didn't think in this direction at that time. Yeah, so this is how we extract DNA. And in Silent Lab, there is a special ancient DNA facility, and they are going to have a big conference on the 29th of November. So if anyone is interested, on the 29th of November in Stockholm, in the Center for Paleogenetics, there will be a big meeting of these guys. So after we extracted DNA, this is how we do sequencing, so-called sequencing. Sequencing means we read this DNA. And the way it's done typically, it's chopped into small pieces. Unfortunately, due to technological limitations, it's impossible to read big stretches of DNA. But you have to chop it and read sort of piece by piece. So we chop it, and then there are special adapters that attach this DNA to some platform. And then there are nucleotides that are added one by one to this platform, and they are sort of attached to this strands of DNA and build a complementary strand. So now it's a single-stranded DNA, and then they add these nucleotides, and nucleotides they attach one by one and build a complementary strand. And every time they attach, they emit a certain color, certain light. And then there are special machines that can read these colors. And this is how so there are only 4 colors because there are 4 nucleotides. And this is how we can read C, T, G, T, T, C, A. We can read this sequence. And then, so this is the alignment process that I briefly explained. So we have a reference genome. So human genome has a reference, and many other organisms have a reference genome. And then these sequences, they came from particular, maybe ancient sample. And then you align them against this reference genome. And sometimes you can see that at certain positions, these query sequences, they're different on one position in comparison with the reference. And this is how you detect mutations. And these mutations, they can tell you something about they're usually linked to some qualitative traits, or our human traits. These mutations, they don't happen. Some of them do not have meaning, but many of them, they actually lead to some changes in our body, so to say. And there are special machines that can read these sequences, and this is what we bioinformaticians analyze. So we are starting working here on this step. So we are looking at the sequences. So what is the peculiarity of ancient DNA? As we already mentioned, it's degraded. It's very fragile. It's very short. And if you sort of plot the distribution, length distribution, so here is a length and here is a frequency, so that you find on average, you can find maybe, I don't know, 40 base pairs segments. They are sort of the most abundant. As I said, 30 is already a threshold of specificity. So that's why 40 is not long. We would like, I mean, when we work with modern DNA, actually we have much longer segments. But ancient DNA has short segments, and there are a lot of troubles because of this. And in addition, so there is a process which is called deamination process. It turns out that there is some weird chemistry going on with time, that there are some enzymes that sort of incorporate some artificial mutations in the DNA, post-mortem mutations. So those mutations, they were not inherited from from the parents of that ancient individual. So those are not true biological mutations, but those mutations happened when the bones were in the soil, and they were sort of degrading, so to say.
What's the time constant of that, roughly? Is it 1,000 years or?
You mean when it happens? So there are different examples. So people even try to make a so-called molecular clock, try to date instead of carbon dating. So carbon dating is a golden standard in ancient DNA, but people try to look at this deamination process and see, okay, if there is a lot of deamination, it's an older sample. The problem is that permafrost samples look like almost modern samples, even though they can be, like carbon dating says they are from 10,000 years ago. But there is very little deamination. So it really depends on preservation conditions. So preservation conditions are actually more important than actual biological age of a sample. So this deamination happens due to basically preservation conditions. But I hope you can see now that these artificial mutations, it's hard to actually discriminate them from the true biological mutations.
But so it's only a problem, it's not an opportunity.
It is an opportunity also. I see it as an opportunity. Not everyone agrees with me, because people usually, in the lab of Louis Dallin, for example, who works with mammoth, they remove damage as a routine. They use special user enzymes that remove damage. So, and this is, I think, I always argue with Louis, sort of, I think it's a crime, but people use it for population genetics analysis. They want robust mutations. And of course, with damage, it's more difficult to call mutations, to be confident that it's a true biological mutation and not this artificial.
If you take
mutation.
a sample and put it in the machine and run it, can you take out the sample and run it again? Or is it destroyed?
Yeah, so usually it's not a good idea to do it this way.
So you need to half the sample and treat half of them your way, half of them.
The thing is that we are surrounded by a myriad of enzymes that can sort of destroy biological molecules in our sample. So it's really thawing and freezing sample is not a good idea. So it's really, and real biologists, when, for example, talking about single cell analysis, real biologists actually told me many times that get scared what kind of manipulations people do in the lab with the cells, because they say, okay, you see some transcriptional pattern you detect from single cell analysis, but it has nothing to do with the native state of the cells, because you violated them. So you do a lot of manipulations, and then of course it's hard to prove that we haven't introduced any biases. We do the best we can, but of course there might be some biases and artifacts due to lab treatment. Yes, and this is how you statistically detect this domination in the analysis. So as I said, Bo, it's an opportunity. So the degradation is actually a method number one to actually separate modern DNA from ancient DNA. So people typically look at this distribution and see, okay, if the DNA is degraded, if DNA is short enough, then it's highly likely that it's ancient. There are other types of DNA, cell-free DNA, for example, in our blood. It's also degraded. It's not ancient, but so I mean there are other types of DNA that can be degraded, not only ancient DNA, but a DNA extracted, after drilling from an ancient sample probably is ancient, demonstrating this kind of degradation is probably ancient. And also you can check sort of these mutations. They usually happen at the end of the short sequences that you read. So in the sequencing, you read DNA stretches, their sequences, and it turns out that these mutations, these artificial mutations, they happen at the ends of the reads, because of some also biological processes, because usually DNA is single-stranded at the ends, and then there's enzymes that introduce these errors into the DNA code. They're easier, it becomes easier for them to attach and introduce this deamination. But sort of effect is that usually people see these mutations at the ends of the reads. So if you now check, if you take your sample, you have a bunch of sequences. Now you can check whether you find C to T and G to A mutations at the ends of the reads more frequently than any other substitution, than any other mutation. So if you see an enrichment of these particular substitutions, not any, there are many. You can check. You can change maybe C to A, C to G, but I'm talking only about this C to T and G to A. So if you see an enrichment of this particular domination, the so-called transition mutations, then it's probably a good indication that it's a proof, basically, that it's an ancient DNA. Yeah, time is running. So what? When should I stop? Because okay, when you're satisfied. Good. Because I want to sort of make it more interesting for you, and I'm talking a lot, but then it doesn't result in showing all slides I prepared. But I hope you still find this discussion interesting. Yes. So then there is a sort of another difficulty with calling. So that's a terminology of slang. Calling mutations, calling variants. So these different mutations they are called genetic variation. And this genetic variation, what is genetic variation? It's a difference compared to the reference. So all the positions that are in your query sample that are different compared to the reference, those are variation, genetic variation. And you can detect this genetic variation. And there are special tools, special programs that can first align these DNA segments to the reference genome, and then they can detect these mutations. So in this particular in this particular case, you have a SNP. It's called single nucleotide polymorphism. So you have different DNA segments coming sort of from two chromosomes, because we humans have a diploid genomes. We have chromosome pairs. One chromosome we inherit from the mother, the other one from the father. And sometimes, or very often, these DNA sequences from the mother and from the father, they are identical. But sometimes they are not at some certain positions. So what you see here is that some certain DNA segments, they came from one chromosome and with allele or nucleotide C, and others they come from another chromosome with a nucleotide A. And this is when you have this, so A and C, different sort of versions of DNA at this specific position. They're called alleles. That's another terminology. And if you have sort of two alleles equally frequent at a certain position, then this position is called heterozygous position. If those two are identical, maybe AA, there will be AA or CC. It's called a homozygous position. But it's still a genetic variation. It just this particular individual is heterozygous and another individual is homozygous. And this AC is called SNP. I think many of you heard this terminology, single nucleotide polymorphism. So the problem, so actually I was not going to talk about heterozygous and homozygous, but just to show you how the ancient DNA alignment looks like compared to modern. So that's a modern DNA. So you have many DNA segments aligned to this region, a reference genome region, and you can reliably sort of detect that this is A to C polymorphism. Because you have you have a lot of evidence. You have many reads. Again, it's terminology. These short DNA sequences that are read during sequencing process are called reads. So these reads, there are many reads covering this position. While here you have maybe 1, 2, 3 reads. So it becomes very difficult to actually detect these mutations when you work with ancient DNA. That's another difficulty. So damage, degradation, low coverage. So this is called low coverage. By coverage, people mean how many, on average, DNA sequences you can get. You can sequence. You can sort of read for that particular position. So here the coverage will be 1, 2, 3, 4, 5, 6, 7, 8 times. 8 times. So you have 8 reads. Here the coverage is 1. That's a terminology again in bioinformatics. Yes, another difficulty is what I already mentioned, is that it's hard to actually discriminate between SNPs due to true biology, due to so-called germline mutations, SNPs that happen, that you inherit from your parents genetically. It's hard to discriminate them from this artificial gemination SNPs. So, for example, here, those are two situations. Both of these SNPs are C to T polymorphism, heterozygous positions. However, here you can check that this T allele, T nucleotide is present at the end of this read, and C is present at the end of this read, and again T is present at the end of this read. So meaning that probably this is highly likely that this was actually a gemination process that introduced this. And this is more reliable because here it looks like these polymorphisms, they're not, they're supported by reads and originate not from the ends of the reads. This is how typically programs that do this variant calling, they function. They actually can check whether this single nucleotide polymorphism, they come from the ends of the reads or not. That's briefly sort of another difficulty, another sort of challenge that people have working with ancient DNA. You don't have this problem with modern DNA. In modern DNA, there is no gemination. So both of these variants would be robust for modern DNA, but not for ancient. And then, of course, if you have 50% of reads due to gemination, for example, what kind of genetic analysis you can do, you can imagine. You can get very wrong results, and in this area, still it's not safe. There are a lot of speculations. So there are a lot of artifacts in this field, unfortunately, because it's very hard to get rid of these constraints.
Is it typical that we use all of the material in analysis, or is it some sort of kept for future studies that if the technology improves? I mean, some of the artifacts might be due to the technology used today, or are you destroying all the material just to get to the most reads as possible?
you do destroy a very unique museum sample. For example, Denisovan, so it's another type of Neanderthal, so to say. Denisovan individual, the first one that Svante found in Denisova cave. So they had a fingertip, a bone from a fingertip, like tiny, like a few millimeters only, and they crushed it all and they extract a lot of DNA. So, and then looking at the DNA, they figured out that this was a very strange Neanderthal. It was like super different from modern humans and also from ancient Neanderthals. That's why they came to this concept of Denisovan and another Neanderthal. Because there was no skull of Denisovan found ever. So people have a lot of skulls of Neanderthals. They can actually see morphological difference between human, anatomically modern humans and Neanderthal skulls. But still, Denisovan was detected purely genetically, based on DNA. People haven't seen skulls or bones of Denisovans.
So if you had the same issue as with the mummy, now the evidence is gone. So you cannot...
It's gone, exactly. Yeah. But happily, people find more and more bones that, like reliably, there is maybe one more Denisovan found, but there are many candidates. So people are still working on it in order to figure out whether it's Denisovan or not. But you're right that sort of you destroy the fingertip, and of course museums, they don't want us to destroy, because we unfortunately need to sacrifice this unique material, and there is always a struggle between sort of administration, museums. They don't want to give us samples for genetic analysis. So a few examples. This was an introduction. I tried to sort of emphasize what kind of challenges we have in ancient DNA, general challenges. Then there are tons of branches. So the ancient DNA branched in different directions, and I'll try to give you some flavor of a few of them. Maybe you'll say, "Wow, that's really cool. I want to read up on this." So the Neanderthal genomics, this is how sort of, this is where this paleogenetics research, at least using next generation sequencing, started with Svante and his group. Nowadays, it became much more than just Neanderthal research. We are talking a lot about Neanderthals because they're interesting, because they're closest species, closest organisms to us. So there are apes, and then there are Neanderthals, who are the closest to human being. That's why they're so fascinating. And if you're not familiar, so there is a hypothesis, so-called out of Africa hypothesis. It's not the only hypothesis, but it's dominating. It's a conventional sort of way to describe history. Modern humans, anatomically modern humans, they migrated out of Africa maybe 50,000, 60,000 years ago, and they met Neanderthals here in Europe, who already lived in Europe and Asia. Denisovans were living in Asia. Where Neanderthals and Denisovans came from, that's a mystery, but probably also from Africa. So the more people work, the more they realize that there were not just a single migration, but there were back and forth migrations. So previously, people thought that you cannot find Neanderthal DNA in Africa. But now there is more and more evidence. People realize more and more that actually, apparently, this was like a highway. People were migrating back and forth. But conventionally, what is written in textbooks is that like 50,000 years ago, people, anatomically modern humans, they migrated out of Africa, and they met Neanderthals, and they had common children. So meaning that we are the same species. We are not separate species. So there was a process called introgression. So Neanderthal DNA was introgressed or admixed to our DNA. And on average, Europeans have 2% of segments from Neanderthal that we sort of can reliably say that they came from Neanderthals. And so Svante's group was the one who sequenced draft Neanderthal genome in 2010. And this was one of the first applications of this next generation sequencing. You remember, you chop DNA, you attach it to the platform, and then you add nucleotides. This is called next generation sequencing. Because
you said in average 2%. What is the max ever seen?
Yeah, I mean, there are some extreme individuals with sort of poor.
We all know who they are.
People from Papua New Guinea, they have 4%. They have 4% of Neanderthal DNA, or Denisovan DNA. So they are most extreme individuals in the world in terms of Neanderthal DNA. But in Europe, it can be almost 0.5%, it can be 6%. There is a variation. Maybe I can show you. Yeah, that's actually this is the confidence intervals of Neanderthal ancestry. So this is a 2%, this is 4%, and here is so these three individuals, they're Africans. So the African individuals from different African populations, they have almost 0% of Neanderthal DNA. But then Han, this is a Chinese, and then French and Sardinian, so those are Europeans, and then this is Papuan. The Papuan they have on average like 4%, but it can be, you see the variation. So the variation.
Sorry, how can it be negative?
Yeah, I mean, confidence,
Confidence is not the distribution of individuals.
Yeah, so if the confidence interval sort of overlaps with a 0, it's meaningless. I mean, the mean can be any, basically. So how exactly this can be negative? I think it comes from this definition. So what is plotted actually here is called D-statistic. So this is called D-statistic analysis. This is how historically, that was the first time Svante and his group here in this paper, 2010, they were using this D-statistic in order to show the presence of Neanderthal introgression in modern human. And it works like this. So that for each position, for each maybe single nucleotide polymorphism, you have 4 populations. And one of them is outgroup, so-called outgroup population, it's apes. And then, during history, so here we had common ancestor with apes, and then Neanderthals split from modern humans. So those two are modern humans, Africans and Europeans. Neanderthals split from modern humans, and here at this point, humans, Africans and Europeans split. So you have 4 populations. But then there are sort of, at each position, you can check 2 alleles, because we have a diploid genome. So meaning that potentially we have 2 different alleles at each position, one from mother and one from father. And you can check actually, so there is a concept of derived allele and ancestral allele. Ancestral allele is the allele that the apes have. It's the old one. And then sometimes Europeans or Neanderthals or Africans, they can have these ancestral alleles, and sometimes they can have a derived allele, the new one. They knew that the apes don't have at all, something that happened during evolution. And then you compare two different scenarios. You compare whether there is an enrichment of shared ancestral alleles between European and Neanderthal, or between African and Neanderthal, in order to figure out whether Africans are closer genetically to Neanderthals, or Europeans are closer genetically to Neanderthals. And so null hypothesis is that the frequencies of these 2 situations are the same. But if there is an enrichment, if you see sort of statistical enrichment, you can sort of test it statistically that this number is significantly higher than this number. Then you say that there is an excess of shared derived alleles between Europeans and Neanderthals. So this is how people figured out that Europeans are much closer to Neanderthals compared to Africans. So Africans, they do not, sure, yeah.
What is an allele?
Yes, the allele. So I was saying that here, I was introducing, I realized that I had to, this was the very last sentences I added, because I realized that, yes, I'm going to use the word allele. And when I started doing bioinformatics, I was asking people, what is allele? What is that? because yeah, indeed, it's a strange terminology. So the allele is sort of it's a copy. It's a version of DNA at this particular segment. And since we have sort of 2 chromosomes when we sort of born, we human individuals born, we inherit 1 chromosome from the mother and 1 chromosome from the father, and they can be identical at all positions, but they can vary sometimes. So your mother's DNA can be different, a little bit different from your father's DNA at certain positions. So these 2 versions of DNA from different chromosomes, those are called alleles. So, but it's sure. Yes.
I was just thinking, I find it super interesting, but I think in the interest of time, maybe we should run up and leave some room for questions.
Yeah.
The people who need to go somewhere and they might have some questions.
I realized this. I'm very sorry. I'm very sorry. Just a very last word about this Neanderthal introgression. So people actually learn how to call segments of Neanderthal introgression. And they sort of, to do this, they use these techniques. So the D statistic was one way to detect Neanderthal introgression, genome-wide, so to say. But then you can specifically check it from which positions in human genome, where exactly you have Neanderthal segments, and whether these specific positions, they overlap with gene regions or intergenic regions, whether there is some functionality related to these positions. And they sort of, the idea to call these segments of Neanderthal introgression is that you compare your test sample, maybe European sample DNA stretch, you detect these positions where here Europeans have A allele and Neanderthal has A allele, but Africans have C. And then it should not be just one position, but there should be a segment, a group of SNPs that inherit together, which is called haplotype, another terminology called haplotype. So the whole haplotype should be, the test sample should have much more genetically closer haplotype to Neanderthal than to African. And then you can, the segment should be not short enough and should not be long enough, because it can be computed when exactly the split from Neanderthals, the Neanderthal introgression happened, when exactly in time. It was like 2,000 generations ago. The Neanderthal introgression happened, and you can compute sort of when these haplotypes, these regions that inherit together, they become shorter and shorter with time. The more generations pass, the shorter these haplotypes become. So these haplotypes should satisfy a certain genetic lengths. So basically, by applying these 3 criteria, people learn how to call it. It's hidden Markov models that who actually check this. So they sort of scan our DNA for these regions. Yeah, I think it's all I wanted to say about Neanderthal introgression. There were questions. Oh, do you guys want to ask questions now? Yes, yeah, you had a question.
I had one question apropos what he asked about the percentage, and it might be you might not be the right person to answer, but to which extent is that percentage relevant? I can understand that it's relevant in order to see how much we have intermingled in the past, but 2% DNA, because DNA doesn't tell you, a lot of repressive genes that doesn't determine phenotypes.
Exactly,
right?
yes.
So,
and I can,
to which extent is a percentage?
Yeah, I can tell you that, yes, people actually checked whether these introgress segments are relevant, where exactly they are, in genic regions or not. So it turns out that they fall into outside of genes, outside of coding regions. So there is a hypothesis. It's not like written in stone, but this is what people believe, that Neanderthal DNA was for some evolutionary reason not beneficial for us. And evolution tried to eliminate this harmful DNA from our genes. So indeed, you're right that Neanderthal DNA is usually concentrated not in genic regions. And if it was sort of just a recent Neanderthal introgression event, then you would expect that Neanderthal DNA would be sort of randomly distributed. Only with evolution, with time, you notice that certain segments, they survive evolution, functional segments, certain they don't survive. Because sort of you give, evolutionarily speaking, you give fewer offsprings, so you propagate your DNA less and less. This is how sort of your weak lineage disappears in terms of evolution. So it's a good question that Neanderthal DNA, there is something strange, there's something wrong evolutionary with Neanderthal DNA. More questions? Sure.
On the same topic, really. So if you do this sequencing back in time, say on graves, maybe from the Middle Ages, or so, can you see this movement of?
Yeah, so you're touching something I wanted to talk about because it's super interesting for you guys. I realized time is your variable of interest. And I specifically prepared a few slides, time series. Can we use sort of time series analysis? Typically, so ancient DNA is a great topic for you, I believe, because it offers naturally time series data. Because if you, for example, compare with medical research, with modern DNA biomedical research, usually they don't have that many years. They have longitudinal data, but usually they work with cross-sectional data. They don't have time as a variable at all. Sometimes they have 3 time points or 4 time points, but this is, you can't do, you know, fast Fourier transform, or you can't do autocorrelation. You can't really work with 4 time points, you know. But in ancient DNA, you have these long trajectories, long trajectories. And this is sort of people noticed that allele, it has a frequency in population. So for example, a nucleotide at certain position is more frequent in Europeans, for example, than in Africans. So that's called allele frequency. In across population, you count how many times you find A at this position. This is called allele frequency. And it turns out that this allele frequency can sort of fluctuate. And sometimes it can go from 0, basically, from 0 in population to 1 in population. And there are two sort of, there is a long-standing debate in evolution. It's very hard by looking at trajectory of this fluctuation of allele frequency. Very hard to say whether this fluctuation is due to selection. So there is a selection. In evolution, people from Darwin, they have this natural selection concept that allele is decreasing in population. The frequency of allele is decreasing or increasing in population. Sometimes for a reason. There is a reason. There is some evolutionary pressure, some climate change, you know, some, I don't know, some demographic effects. People are migrating to different countries. There are sort of different obstacles that can different ways we can influence this fluctuation of allele frequency. And so this is, and then the question is, when you see the kind of trajectory, is it purely random? Is it random walk? Is it white noise? Or there is some sort of some correlation here. There is some memory here. And if you detect this memory, so usually this is a selection trajectory. Basically, if you in simulation, it can be easily shown that sort of if you apply some evolutionary pressure, then this trajectory, allele frequency trajectory, it follows it follows like stepwise function, while genetic drift, and this is in mathematical terminology, this is random walk. So evolutionary people, they call random walk, they call it for genetic drift. This genetic drift, it can also go from 0 due to genetic drift. Allele can go from 0 frequency to 1 frequency, exactly like here, but the trajectory will be very different. And then by looking at autocorrelation functions and the derivatives of of these trajectories, because yeah, I was going to talk about a little bit, but maybe next time. So basically, that's something. The summary, to Christian, just to answer your question. Time series data you will find a lot in ancient DNA, and the topic they're interested in is actually to detect selection. to detect natural selection. And the challenge here is actually to prove that allele frequency changes not randomly. So the random change is a null hypothesis. You have to kind of do some analysis in order to prove that this decrease in allele frequency, maybe it doesn't mean anything, just you know, by chance. But sometimes it's not. Sometimes there is an evolutionary pressure, and that's exactly the question they want to answer. Yes.
I have a question. So let's take two genome sequences, one from one individual and one from another individual.
Mm-hmm.
Can you say, like, quantify, like, how different are those two? Like, is there like a genome calculus, like A minus B, something like how different is these two genomes?
Yeah, sure. So that's actually what the phylogenetic analysis is doing. In evolutionary research, there is a sort of a concept of phylogeny. So when you construct this hierarchical dendrograms, so you can call it like people with mathematical background would say that this is hierarchical clustering. So that you just the underlying data here is genetic data, meaning that you sort of count how many times chimp alleles are identical to Neanderthal alleles and modern human alleles. So you go from one position to another and you count the differences, pairwise differences. You count how many times chimp is different from human, how many times Neanderthal is different from modern human. And then this is how you construct this tree. So that you learn that some samples are closer genetically to each other, just looking at pairwise differences. So maybe they're totally identical. There's two genomes.
It should also be that both the position and frequency should matter, right?
Mm-hmm. So here we are talking about sort of there is a population analysis. Sort of here, imagine, so here there is one chimp, one Neanderthal, and one modern human. But imagine you have a group of chimps. They still, if you add them to your data, they still will be clustering here, but sort of also they will not be identical. So the allele frequency that I mentioned that fluctuates, the trajectory of allele frequency that fluctuates, this is a population allele frequency. So you compute this across individuals from the same population. But if you compare different organisms, you can still well, I mean, if you're interested in similar dendrogram for only humans, then yes, you can detect that Africans, Europeans, Asians, maybe they cluster, they form separate branches on this tree. But this is essentially what you're doing is called phylogenetic analysis. And again, in mathematical term, this is just dendrogram hierarchical clustering. But the problem here is that the ones who did hierarchical clustering, for you, a warning is that it turns out how phylogeny is different from hierarchical clustering is that they actually, during time, they have backward mutations. So for example, sometimes C mutates to T, but then it mutates back to C. So you actually see, you don't see some positions as mutations, but you can still account for this effect. It's called saturation effect. That's very, very different from traditional hierarchical clustering analysis. So you have to account. So basically the phylogeny is essentially about accounting for this saturation effect. You have to account for these back mutations. So C mutates to T and then to A and then back to C, for example. You don't see it. So you observe an individual that has C. And you say there is no mutation here because it's C and the reference has C. But actually, in fact, during evolutionary process, this position, it experienced mutations from C to T and then to A and then back to C. That's why right now you don't see it as a mutation, but actually it happened. And these mathematical models that actually construct these phylogenetic trees, they try to account this back mutations or so-called saturation effect. More questions?
I think there are a lot, but I also
I'm sorry. I can come again.
We should definitely keep in touch. And I hope that
I want to hear no more.
Mm.
Wouldn't we formally end the seminar and then?
Exactly, yes, yes.
The
Whoever wants to.
Yeah, the ones who want to continue, I think I still have strength.
So first of all, we would like to thank you for this
Yeah.
presentation.
My computer is going to die.
and then we continue.
Yeah.
Thank you for your questions.
I have one question. So, for some of the DNA, you can follow the paternal line. There are some DNAs where you can follow the maternal lines. Is this used in the field to?
Yeah. So you're talking about X chromosome and mitochondrial DNA. The mitochondrial DNA, for example, is inherited only from your mother. And this is how people, sort of, the advantage of mitochondrial DNA is that it's very abundant. So basically, it turns out, so mitochondria has its own genome.
Yeah.
So there is a nuclear, there is a like a normal DNA in our nuclei, in our cells, but then there is